Single-cell electroporation is a specialized technique allowing the delivery of DNA or other macromolecules into individual cells within intact tissue, including in vivo preparations. When combined with advanced in vivo imaging techniques, single-cell electroporation of fluorescent markers permits direct visualization of cellular morphology, cell growth, and intracellular events over timescales ranging from seconds to days. In this video, we detail the procedure for single-cell electroporation of a fluorescent dye or plasmid DNA into neurons within the intact brain of the Xenopus tadpole, and show examples of live imaging of fluorescently labeled neurons.
Oh, hi. My name is Sesath Hewapathirane, and I work in the laboratory of Dr. Kurt Haas in the Brain Research Center at the University of British Columbia. Today, I will demonstrate how to perform targeted single-cell electroporation of neurons within the intact brain of the albino Xenopus laevis tadpole. The distinct advantage of this technique is that experimental manipulations may be performed on individual cells while leaving the surrounding tissue unaltered, thereby distinguishing cell autonomous effects from those resulting from global treatments. The first step involves fabricating micropipettes with appropriate tip diameter and taper angle, followed by loading transfer material into the micropipette. Then, tadpoles must be anesthetized and transferred to the electroporation chamber, followed by inserting the micropipette into the brain and applying a train of voltage pulses. Finally, electroporated neurons are imaged using 2-photon microscopy. Though I'll be showing you this technique in tadpoles, single-cell